Hello everyone, welcome to my YouTube channel. Today I am going to talk about mitosis and cell cycle. If you are asked in viva regarding what is mitosis, whether in dysplasia has mitosis, what is cell cycle, and lastly, how does mitosis look in the microscope? I hope in this video you will find an answer to those questions. Before we start, there is a disclaimer, and the disclaimer is the same in all my videos. Thank you. Now let's begin. What is mitosis? Mitosis is the process in which a eukaryotic cell nucleus splits into two, followed by division of the parent cell into two daughter cells. In the context of cell cycle, mitosis is the part of the division process in which the DNA of the cell nucleus is split into two equal sets of chromosomes. What is the purpose of mitosis? In A, regarding development and growth, mitosis populates organism body with cells. In B, mitosis replaces old, worn-out cells with new ones. In C, the purpose in mitosis is it acts as a form of reproduction, adding new individuals to the population. So what is the goal of mitosis? The goal is to make sure that each daughter cell gets a perfect full set of chromosomes. Cells with too few or too many chromosomes usually don't function well. They may not survive, or they may even cause cancer. If not corrected in time by the body, the mistakes made during the mitosis can result in the changes in the DNA that will lead to genetic disorder. With the genetic disorder, this will predispose to formation of dysplasia. And this will answer the question regarding whether in dysplasia has mitosis or not. What is cell cycle? A cell cycle is a sequence of events that a cell goes through as it grows and divides to produce new cells. Therefore, it can be called the life cycle of a cell. Before a dividing cell enters mitosis, it undergoes a period of growth called interphase. 90% of cell's time is in the normal cell cycles is in the interphase, as illustrated by the picture on the right-hand side. In G1 phase, it is a period prior to the synthesis of DNA. In this phase, the cell increases in mass in preparation for cell division. The G1 phase is the first gap phase. The S phase. It is the period during which DNA is synthesized. In most cells, there is a narrow window of time during which DNA is synthesized. The S stands for synthesis. The G2 phase. It is the period after DNA synthesis has occurred but prior to the mitosis stage or prior to the start of prophase. The cell will synthesize protein and continue to increase in size. The G2 phase is the second gap phase. In the later part of interphase, the cells still have nucleoli present. The nucleus is bound by a nuclear envelope and the cell chromosomes have duplicated but are in the form of chromatin. What are the phases of mitosis? Some describe as 5 phases, whereby after the prophase, there is a late prophase phase, or they call it Metaphase. But I will describe it based on 4 distinct phases. Number 1 phase is prophase. Number 2 phase is metaphase. Number 3 phase is anaphase. And number 4 is telophase. In each phase, there are characteristic steps in the process of chromosome alignment and separation. Once the mitosis process is complete, the entire cells will be divided into two by the process called cytokinesis. In number 1, prophase phase, the chromatin condenses into discrete chromosomes. The nuclear envelope breaks down and spindle form at the opposite poles of the cells. Prophase phase is the first step of mitotic process. During prophase, the chromosome condense into a X-shaped structures that can be easily seen under a microscope. In prophase, each of the chromosome is composed of two sister chromatids that is attached by a centromere. Each chromosome contain identical genetic information. In prophase, the nuclear membrane breaks down. Centrioles will move toward the opposite ends or pole of the cell. For the second phase, known as metaphase, the chromosome will line up in the middle or the equator of the cell. The spindle fibers from the centrioles attach to the centromeres of every chromosome. For the third phase, known as anaphase, the paired chromosomes or sister chromatids separates and begin moving to the opposite ends or poles of the cell. The spindle fibers not connected to chromatids begins to lengthen and elongate the cell. At the end of anaphase, each pole contains a complete compilation of chromosomes. For number 4, or last phase, also known as telophase, the polar fiber continue to lengthen. Nuclei will form at the opposite poles. The nuclear envelopes of these nuclei form from remnant piece of the parent cell's nuclear envelope and from pieces of the endomembrane system. Later, the nuclei will reappear. The chromatin fibers of chromosome will uncoil. Telophase phase is now complete, whereby the genetic contents of one cell is divided equally into two cells. What is cytokinesis? Cytokinesis is the physical process that finally splits the parent cell into two identical daughter cells. During cytokinesis, the cell membrane pinches at the cell equator, forming a cleft called the cleavage furrow. The position of the furrow depends on the position of the astral and interpolar microtubules during anaphase. The cleavage furrow forms because of the action of a contractile ring of overlapping actin and myosin filaments. As the actin and myosin filaments move past each other, the contractile ring becomes smaller, akin to pulling a drawstring at the top of a purse. When the ring reaches its smallest point, the cleavage furrow completely bisects the cell at its center, resulting in two separate daughter cells of equal size. In conclusion, mitosis is the process of nuclear division which occurs just prior to cell division or cytokinesis. During this multi-step process, cell chromosome condense and the spindle assembles. The duplicated chromosomes then attach to the spindle, align at the cell equator, and move apart as the spindle microtubules retreat toward the opposite poles of the cells. Each set of chromosomes is then surrounded by a nuclear membrane, and the parent cell splits into two complete daughter cells. Okay, that's all for today. Sorry for the mistake in a few pronunciation, but I do hope that you have learned something from this video. See you next time. Bye.